My fellow interviewer and I are excited to present to viewers a never-before-seen exclusive Zoom interview of the current top runners for the organelle office. Please welcome presidential nominee Mitochondria and his running mate, Chloroplast.
Hello, I'm Mitochondria.
Hey there, I'm Chloroplast.
So, let's get started. Mr. Mitochondria, where are you from?
Well, I travel a lot, but I'm definitely in the cytoplasm of the cell with my pal, Mr. Chloroplast.
Very interesting. How would you describe how you're built?
Well, I'm very structurally sound. I'm enclosed by two phospholipid bilayer membranes. The outer membrane is where proteins can pass into or out of mitochondria, and the inner membrane contains the proteins used during ATP synthesis. I also have an intermembrane space that is the region between those two membranes, and a matrix enclosed by the inner membrane that contains my enzymes, my DNA, and my ribosomes.
How about you, Mr. Chloroplast?
Well, I have three compartments: an intermembrane space between the inner and outer membranes, a fluid stroma between the inner membrane and glycolides that contains my DNA, ribosomes and enzymes, and a glycolide space enclosed by the glycolide membrane.
Our next question is, what do you even do? Like, what do you use and provide for the cell?
Well, I'm simply the site of cellular respiration, which is the process that generates and provides energy in the form of ATP that drives most of the processes in the cell. I use the sugar glucose, derived from the food we eat, and oxygen to produce this energy. You may ask how I do this. Well, I'm very versatile. I use the energy I gain from the electron transfer chain from my inner membrane when electrons move from molecule to molecule due to oxidation to maintain a proton concentration gradient between my intermembrane space and my matrix. As the protons travel down the gradient in a process called chemiosmosis, this drives ATP synthesis in enzymes in my inner membrane called ATP synthase. This process of ATP synthesis is called oxidative phosphorylation.
And you, Mr. Chloroplast, what do you do?
I'm the site of photosynthesis, where I convert solar energy to chemical energy by absorbing sunlight and use it to synthesize sugars like glucose, which the cell can subsequently use as energy to carry out their life processes. I use light energy along with carbon dioxide and water to produce glucose as an energy source. I do this through two phases. My light reactions occur at the glycolide membrane, where solar energy is used to synthesize ATP and NADH, and my Kelvin cycle occurs in the stroma, which uses the previous products along with carbon dioxide to synthesize sugars for the cell.
And what about those that depend on you?
Since me and Mr. Chloroplast are responsible for providing energy for the cell, many of the other organelles depend on us for supplying them the energy to carry out their own functions. For instance, ribosomes use the ATP we make while they are doing protein synthesis. The Golgi apparatus needs ATP to modify contents of transfer vesicles and send them to other destinations. I even personally work with the ER to coordinate cellular biological functions such as calcium ion signaling, apoptosis regulation, and even the ER stress response.
That's very cool. Now, what would you say are the biggest reasons that voters should vote for you?
Well, since the cell gets its energy from us, the cell couldn't carry out many of its life processes without us. The photosynthesis I do is necessary for much of life, since plants are one of the most important producers, and many heterotrophs use the energy stored in the plants to carry out their own life processes, as they break down those sugars through cellular respiration with mitochondria. Without us, the cell would have no energy to live.
For real. And not only do I generate energy in the form of ATP, I also store calcium in order to regulate cellular metabolism. In addition to stimulating ATP production, the regulation of calcium involves cell signaling of other processes, including apoptosis in which the cell is destroyed, and the regulation of calcium is so essential for cell life and death.
If you need a third reason, the byproducts of photosynthesis and cellular respiration are necessary for life. For instance, excess oxygen is released during photosynthesis, which most organisms need to breathe. Also, carbon dioxide is released as waste during cellular respiration, which plants need in order to survive. It's a win-win for everybody.
And why are the both of you so much better than your competition?
Oh, that's easy. Well, you see, abnormalities in the cytoskeleton, such as malfunctioning microtubules and other microstructures, have been shown to lead to cardiovascular disease syndromes, neurodegeneration, cancer, liver cirrhosis, pulmonary fibrosis, blistering skin diseases, Parkinson's disease, Alzheimer's disease, Huntington's disease, amyotrophic lateral sclerosis, etc. Do you really want a deformed and fragile leader?
Don't even get me started with prokaryotes. They have been shown to be responsible for many diseases, since they have fewer regulations, which allows for rapid mutation and multiplication. Some bacterial diseases include cholera, diphtheria, bacterial meningitis, tetanus, Lyme disease, gonorrhea, and even syphilis. Even incredibly deadly diseases like the bubonic plague and MRSA infections, which kill millions of people. Do you really want these prokaryotic menaces in the office?
And even on top of that, the cell membrane and its proteins are just as bad. We trust them as our first lines of defense against viruses, bacteria, and other harmful toxins, but they manage to even fail at that sometimes too. Viruses can trick the cell membrane cell receptors and attach to them, subsequently injecting their viral information and hijacking the cell. Electing the cell membrane and proteins would be a matter of national security.
Well, you heard it here, folks. Straight from Mr. Mitochondria and Chloroplast themselves. So, do your part and let's energize the presidency. Vote Mitochondria and Chloroplast today.